Children, you have to study enzyme inhibitors. So, what are enzyme inhibitors? The substance which stop the activity of enzymes are called enzyme inhibitors. These substances interact with the enzyme, and after interacting with the enzyme, they stop the activity of enzymes, or in other way we can say the enzyme lose catalytic efficiency. So when the catalytic efficiency of enzyme is lost, enzyme is not able to catalyze the chemical reaction. These substances change the structure of molecule of enzyme. Therefore, enzyme molecules lose the catalytic properties. Enzyme inhibitors are two types. There are two types of enzyme inhibitors. First is competitive, and the second is non-competitive. So, which are the competitive inhibitors? Competitive inhibitors have structure similar to the substrate molecules. Therefore, they compete for the same site on the enzyme molecules. The site which is occupied by normal substrate molecules, that is occupied by the inhibitor molecules, because the molecules of inhibitors have the structure like to the substrate molecules. Therefore, they will form to combine with enzyme inhibitor complex, and when enzyme inhibitor complex is formed, it is irreversible, and therefore after the formation of enzyme inhibitor complex, enzyme cannot catalyze the chemical reaction. Generally, substrate molecules combine with the active site of enzyme molecules and form enzyme substrate complex, and after that enzyme substrate complex break and products are produced or products are formed. But here, instead of the substrate molecules of the inhibitors whose structure is similar to the substrate molecules has attached to the molecules of enzyme. There is one example. This will help you to understand how competitive inhibitors are working. See, there is an enzyme that is succinic dehydrogenase. This enzyme changes succinic acid to fumaric acid. Succinic acid combine to the enzyme molecule succinic dehydrogenase and form enzyme substrate complex. After that, it will give product fumaric acid. But there is an inhibitor, that is malonic acid, and structure of malonic acid is similar to the succinic acid. So when malonic acid is present, at that time Maluric acid combine with succinic dehydrogenase enzymes and it will form enzyme inhibitor complex and it is irreversible. Therefore, now substrate molecules, that is the succinic acid, that cannot combine with the succinic dehydrogenase, and therefore now this succinic dehydrogenase enzymes cannot catalyze the reaction. Next is non-competitive inhibitors. Non-competitive inhibitors do not have any structural similarity with the substrate. You have studied competitive inhibitors have structural similarity with the substrate, but non-competitive inhibitors are not having any structural similarity. So they do not compete with the substrate for active site. But non-competitive inhibitors inhibit the reaction. They change the active site. They will attach to some other place on the enzyme molecules, and after that active site of the enzyme molecules will change. And when the active site of the enzyme molecules will change, then substrate molecules cannot combine with the enzyme. In this way, it will stop the activity of enzyme. So there is cyanide. Cyanide has no structural similarity with the substrate binding site of enzyme, but by inhibiting the cytochrome oxidase, which are essential for cellular respiration, it will stop the catalytic reaction.